Am I able to start, Joe?
Yeah, go ahead.
Can you all see my screen? All right, great. Well, thank you everyone for joining our meeting today. It's a pleasure to be presenting an update on the SCORCH consortium, and today's sessions are talking about single-cell approaches to addiction and effects of HIV on the brain. I chair the steering committee with Howard, with Owen, and with great help from John Satterley, who's the Division of Neurosciences and Behavior Special Assistant to the DNB Director at NIDA. So the goal of the SCORCH consortium really is to assay mRNAs from single nuclei. This is sort of a process through autopsy tissues and tissue dissections from animals to isolate nuclei, and then conduct a series of studies to begin to interrogate the sequences within the DNA from each of the nuclei using a number of tools. And the hope is that, as you can see with the depiction here, each nuclei is a single dot, and we will have a number of cell types that we will be able to identify both transcriptionally but also epigenomically information from these different cell types and the different numbers within different brain regions. It's important to note that one of the goals of the SCORCH consortium is to try to understand the transcriptomic and epigenomics of each individual brain cell and how they differ in regard to drug exposure and with regard to HIV, antiretroviral therapy. What's exciting is the data being generated from these different exposures to antiretroviral therapy and HIV being generated through 15 data generating centers that'll coalesce with a SCORCH data center and hopefully provide information to the scientific community and amongst each of the generating centers to develop new mechanisms of substance use disorder and interactions with HIV. The hope is we'll be able to develop novel targets to mitigate the consequences of substance use disorder and HIV with therapeutics to target that. So the question is, how do these brain regions differ? And what is conserved between the different species of human and non-human and animal model systems. To date, with these 15 SCORCH data generating centers, this is an overview of where we are in 2023, with these 15 sites spread across the country and from, as you can see, from Nell Institute all the way down to Cornell on the East Coast. And the program has grown over the years with different substance use targets, different animal models, and different techniques that are being used. So I'll just give you an overview of some of those approaches. In the next slide, you can see the different substances that are being targeted by the different groups that are investigating substance use in HIV, the different types of tissues that are being accessed by the different groups, viruses, and also the techniques that are being used. And these have, as part of the mechanism, are quite flexible and adaptable, but these are sort of the original stated goals and milestones that are being achieved by all of the investigators in the program. Again, just to sort of target the different components of the brain, it's hard to have one individual group target the entire region, but this is sort of an overview of the different groups in the brain regions that are being investigated, but also the source of some of these tissues, particularly from the human brain. We've really been fortunate to have the ability to interact with many individuals, but also many biobanks to be able to obtain these brain region sections for conducting the single-cell studies. So what are the accomplishments that the SCORCH has achieved to date? We've had several working groups that have been put together. There's a tissue and assays group that coordinate discussions and access regarding tissues and assays. We're fortunate that a supplement through NIDA has been provided to members of the consortium to perform toxicology and genotyping assays on all of the human brain samples that are being evaluated by the different groups. And there's been some initial data quality metrics for the single nuclear RNA-seq processes. There was also a data coordination and analysis group that was set up to try to draft metadata and data standards and internal data sharing policies, which, if I understand, has now been achieved. And we're now working on external data sharing policies and have in place now a publication policy that continues to be adapted across the consortium. We have very robust and vibrant monthly steering committee meetings. We have integrated our monthly operational working group meetings now, twice monthly, and we have multiple topic working groups, which I'll talk about, and we'll hear more about these individual working groups' processes and successes over the ensuing years and in the future. To date, there are 50 publications from the SCORCH projects, and 15 of these comprise single-cell data analysis methods. And we have a market paper, which is currently, if I understand, in review, where we'll sort of put together the goals and objectives of the SCORCH program and the progress that has been made to date. The NEMO SCORCH web portal is now up and running, thanks to members of the Data Coordinating Center. This is sort of an overview of the number of files that have been uploaded, the different organisms and projects, and this has been a great platform for us to compile all the data that's being generated and then have visibility across progress in terms of achieving the milestones of the individual consortiums. It's very important that we have visibility across the groups, and this platform, I think, we'll hear more about the data that's been generated, but there's an opportunity to survey what the SCORCH, at least donor data looks like. I think with over 6,000 human organism files, large representation of between both males and females, 309 human donors to date, with 37 of those had deaths from overdose, and these are sort of the diagnoses of those donors in terms of substance use, and the anatomical sites and the tissue sample numbers that have been deposited and have data on right now. So I think it's exciting to see this. If we were to reference the data a year ago, it's incredibly substantial, and it should continue to grow in the years to come. Here's some further analysis of the human data, the distribution of the donors by race and by age, but also HIV status, and also years of HIV infection, quite variable. So there's a wealth of variability in the data in terms of representation of donors across different demographics and characteristics. There is active now 4 task forces within the consortium that meet regularly, and you'll hear a little bit about each of their status and future directions. One focus on the nucleus accumbens, prefrontal cortex, the non-human primate task force, and the viral reference task force, which are really key to consolidate some of the analysis and data metrics that are being put together in guiding these 2 brain regions of interest from both the HIV and the substance use angle. The timeline of activities varies based on the award dates for these individual consortium grants. This is an overview of the onset of the data center initiating as the first program, and then culminating in different grants that support the consortium based on different substances, but also, as I mentioned, different brain regions, but also different animal models and human and non-human primate or rat or mouse models. And this is the timeline to date of all of these programs. So how does SCORCH differ from other single cell projects? There's obviously the Human Cell Atlas, the Brain Initiatives, and then the Human Biomolecular Atlas programs. I think there is some uniqueness to SCORCH in that it is really designed to look at HIV and substance use and trying the best way to identify genes of interest in terms of the data that's being generated. But I think all of these are useful in that we have interactions, mainly through the Data Coordinating Center, to at least be aware of all the other projects and how we can all work together and support the goals of the SCORCH program. So it's interesting that now that we have a plethora of data, it's exciting that the NIDA through John's program at NIDA is now going to support data mining of single cell data and functional validation studies through these 2 RFAs that have been now released. You can use the QR codes here to access these links with the goal of accessing data across SCORCH, but also across the other databases, and really work with a focus on HIV and substance use as part of the objectives of the aims, and sort of the funding that going into both the R01 and R21s that are due middle of next year. So this is a welcome addition to a program to support all the data that's being generated. To date, there has been a number of projects funded, as I mentioned, 15 projects so far with SCORCH, with the RFA DA2305 on large-scale mapping. This is now culminated in 12 R01 projects. And in terms of data mining and functionality, validation studies, these 2 new projects are now moving forward. So I think I'm happy to take questions if there are on these RFAs, but I know John's on the line, they'd be happy to answer questions as well through the meeting. So in terms of the to-do list for SCORCH, I mean, obviously the key metrics is to continue to generate more data. As I mentioned, we have 4 current task forces, and we continue to meet, and I think tomorrow's internal meeting will continue to strengthen the data that's being put together in the analysis and try to align the human metadata from the different brain banks, but also we have the unique capacity to look at cross-species data analysis and then optimize ways to identify these differentially expressed genes across infected and uninfected tissues. The overall goal is to make the data public, and we are working towards achieving that goal going forward. There's a Q&A question. I'll respond to you, Norm, in a minute. Yeah, so what are the current and emerging issues for SCORCH? The reproducibility is a challenge. So same region being processed by different sites. How can we resolve that? And also because we have multiple sites and methods, we've been discussing how to harmonize and ensure we can have reproducibility. The HIV infection and its treatment modalities certainly impact disease measures, and we have additional specimens of plasma and CSF and diagnostic information that would help us resolve some of these issues, and obviously different treatment regimens as well. In terms of substance use disorders, some of the discussions we have on definition, chronicity, single and multiple substance uses, and the temporal relationships into autopsy and end-of-life therapeutics are really key discussion points and continue to be key discussion points in terms of emerging issues that we regularly discuss and help resolve. That would be valuable as the data analysis and moves forward. So with that, I'm happy to take any questions in the Q&A. I see Norm has a question. I think I've responded to the data sharing guidelines that are being worked on for external investigators to access, and that is a key discussion among the group. We anticipate at some point in 2024 that would be realized.
Any other questions for Lish? We still have time.